Hello, my name is Charles Zona, and I would like to welcome everyone to today's McCrone Group webinar. Our presenter is Andrea Champagne of McCrone Associates. Andrea is going to talk to us about good sample preparation and the challenges that different matrices present when trying to do particle isolation. The title of her talk is The Matrix Challenge, Picking Your Problem Cleanly. But before we get started, I'll give you a bit of Andrea's professional background. Andrea is a research chemist at McCrone Associates, specializing in small particle analysis using light microscopy, micro FTIR spectroscopy, and gas and liquid chromatography. Prior to joining McCrone Associates, Andrea worked as a method development chemist in the nutraceutical industry, developing methods to confirm the identity and purity of natural product raw ingredients. Andrea also spent time working as a forensic scientist specializing in controlled substance analysis, fire debris analysis, and clandestine laboratory response. Andrea will field questions from the audience immediately following today's presentation. And this webinar is being recorded and will be available on the McCrone Group website under the Webinars tab. And now I'll hand the program over to Andrea.
Thank you so much. Good afternoon. Thank you for taking time from your schedules today to join us while we discuss some different matrices and the challenges they each present to contaminant analysis. We're going to dive right in, if my screen will respond. What do we mean by the matrix? Well, we're not talking about a special effects laden movie from the 90s. What we're talking about are the components of your sample other than the analytes of interest. In the instance of contaminant analysis, we're talking about the solution or powder that those little black specks have shown up in. Also, too, your collection media can contribute and become part of your matrix. And this happens quite a bit, but it's often overlooked. So we're going to discuss that a little later on and give you some examples of how you're sampling your material should be considered as part of your matrix. But ultimately, you know what your matrix is. It's supposed to be there. The contaminant's what's not supposed to be there. So the matrix is the stuff you don't care about, except you really do care about it. And we're going to explain when and why. Some of the most common matrices that we see here at the McCrone Group are powders or tablets and capsules. So in the instance of a powder that has these little black specks, this is going to be fairly easy for us to isolate. We're going to examine that sample under our microscope. We're going to measure the particles, go in with a tungsten probe, and manually retrieve them. If they're big enough, we can even retrieve them with a pair of forceps. And for a powder like the one you can see on your screen with these nice big clean crystalline morphology, not a lot of that is going to go with our particles, and we'll be able to manually and physically isolate those. So we don't have to worry too much about the matrix in that case. When we get into the finer powders and the tablets and the capsules, now we do have to be a lot more concerned about our matrix. We see a lot of tablets that have either discrete particles in them, where we can shave away some of that tablet material and pull out a whole chunk of something. Other times we see smears or stains on the surface. We're going to do the same thing. We're going to go in with our tungsten probe and we're going to remove the tablet material and isolate just that stained portion. But with fine powders, it's a lot harder to completely remove the matrix. So in that case, we're going to go to a clean area of the tablet or some unstained powder. We're going to take that sample and prepare it for our analysis as well. The most common analyses that we do here are micro FTIR, as we mentioned earlier, or SEM, EDS, scanning electron microscopy with wavelength dispersive spectrometry. Those are our two main identification techniques. We want to be able to attribute any responses from those instruments that are to the matrix and not the particle appropriately, which is why we're always going to sample the matrix when we can. Another very common matrix that we see here are liquids, and this can be any kind of liquid from water to active pharmaceutical products and anything in between. And once again, if we have a large enough particle, we'll be able to go in and manually retrieve that with a tungsten probe or forceps. Sometimes it'll be easier for us to isolate the particles from a liquid solution by filtering. What we'll do in that case is we'll filter the solution through a submicron pore size filter. We'll rinse the container, we'll rinse the filter body, examine it under a microscope, and retrieve our particles from the filter surface. This can be a lot easier to retrieve particles, but it can sometimes get you a little bit more than you bargained for. You might have been looking for this nice big chunk of gray material that looks like rubber stopper, and now we're seeing all of this amorphous, stringy particulate in small white pieces. We need to determine whether that came from the matrix itself, that liquid solution that we originally removed, or perhaps from the container that the liquid was put in. In these instances, we'll always take a portion of the liquid and dry it down so that it can also be prepared for analysis afterwards to determine what should have been present, like maybe that stringy particulate is protein residue, and what is truly foreign to the sample. I'm going to pause for just 1 minute. I've mentioned several times already that we do a lot of manual isolation, and there's a reason that we like to do this. The whole process of visually examining those, physically picking the pieces out, this gives us some truly invaluable information. The appearance of the particulate, whether it's fine crystalline or a shiny metallic appearing particle, does it look like torn plastic? Is there cellular features that might suggest to us this came from a botanical source? All of this is going to be information we can use to inform our decision, our final conclusions for results after the instrumental analysis. The manual preparation of particles also gives us a lot of information about the texture of your sample. Is that black particle elastomeric? When I press on it, does it stretch out and then kind of spring back like a rubber or elastic? Or does it just fall apart into a lot of littler pieces? Is it really brittle? If it does fall apart, how much pressure do I have to put on it to get it to fall apart? All of this is going to give me valuable information later on. Silicone can take a dozen different appearances, and if I have an IR spectrum that shows me silicone and my sample had kind of an elastomeric texture, that's going to indicate to me it's probably a silicone rubber. If it's smeared out like an oil or a grease, it's probably going to be silicone oil. This is why the manual manipulation of those samples can tell us so much. Here at McCrone, we're very fortunate. We have ISO classified clean rooms with some amazing research microscopists who are good enough they can manually manipulate particles down to 1 micron in size. So you don't have to be able to see it with your naked eye in order to gain this valuable information. You just need to have the skills to manually move it. So let's talk about some specific matrices and the specific challenges that they present. In this example, the question I'm asking here is we have little dark spots. Are they adhered to the surface of my material, or are they actually embedded within? Well, if they're adhered to the surface, they're just kind of resting on the top of it, the matrix isn't interfering, and the matrix isn't going to become a concern for us. We're just going to be able to manually retrieve those particles, prepare them for our analysis, and give you a firm conclusion. If it's embedded, it's going to take a little bit more work. We're going to have to remove the matrix slowly and carefully. We're going to, in a lot of instances, the best way to describe it is sort of shaving it off in small portions until we get just to the sample that we're interested in. We'll remove that sample, clear away any matrix that's remaining on it, and then prepare the sample for analysis. In this case especially, the matrix also needs to be prepared for analysis. And if you have a dark spot embedded in nylon tubing, what you really need to do for your matrix analysis there is take a clean piece of nylon tubing and prepare it for analysis. If you have something like food packaging, where something that looks very thin can have multiple layers, take, for instance, a Reese's peanut butter cup wrapper, which is really thin, but actually has between 6 and 7 layers of different polymers. We're going to need to take samples from multiple layers to determine whether or not the sample is truly foreign, or maybe just a piece of top layer brown printing that got trapped in the middle. In a case like that, preparing your matrix is actually going to take longer than preparing your sample. But you need to consider where it came from and all the potential sources of your matrix. Another challenge can be aggregates. Aggregates aren't a single discrete particle. They're more like a loose clump, for technical terminology, of multiple different things. You can see here we've got some discolored powders, some shiny crystalline type materials. There might even be little plastic or metal fragments buried in the middle of that. So this aggregate itself is going to be multiple different types of particles. It can be different types of materials. And sometimes, actually very frequently, your matrix is part of that. So whatever material this aggregate was retrieved from also needs to be sampled and analyzed with the aggregate so that we know which parts can be attributed to the matrix and which parts are truly foreign. Oftentimes, too, you'll see that maybe these brown areas are just melted or burned matrix, so we need to consider the condition of the matrix at all stages. I'm going to talk for a minute now about what I like to call the lingering matrices. When I mentioned earlier filtering some liquids, there are certain liquids and components that just really like to stick around. When we fish a particle out of a liquid matrix, we're going to rinse it off and wash away any of that lingering liquid. Obviously, we don't want it contributing too much. There are some materials, though, silicone, which I mentioned earlier, and protein, which we might find in cellular-based drug therapies, which just really like to hang out and stay on your particles. So we'll wash these off with appropriate solvents. In the case of silicone, we'll use hexane or nonane. And you can see here from the image on the right just how much oil can be retrieved from a particle that looks like it was clean to start with. If you get a particle that has been previously isolated from a liquid matrix and you don't have it available to dry down and examine, it's really important to find out what that was and what it might be, because then you can determine the appropriate solvent to get rid of it. We like to start with water because we want to preserve it. We don't want to risk dissolving our particle, but in the case of silicone, that's not going to take everything off. So as much information as you can have about where a particle originated, the better off you're going to be. It will guide what you're going to do to prepare it to give you the cleanest analysis possible. With challenge number 4 is when I start talking about some of those introduced matrices. Swabs are a great sampling device. You can cover a whole big surface area with one swab. You can get into little tiny tubes and small pipe openings. And if your swab surface picks up discrete particles, it's basically like looking at a filter surface. You can go in, you can manually retrieve those particles, you can size them, and then you can go on with your analysis. If instead of picking up discrete particles, you're getting more like a stain or a residue, something that's soaking into the fabric of that swab, you're going to have to approach that a little differently. In the case of a stained swab, what we're going to need to do is remove portions of the swab that are stained. And if it's a fabric swab, maybe a nylon, we can cut out little discrete portions. For cotton swabs like a Q-tip, we'll tease out individual fibers. And then we'll extract those stained areas in an appropriate solvent. When you're dealing with true unknowns, an appropriate solvent can sometimes mean multiple solvents, where you're looking at perhaps a range of polarities to make sure you're retrieving any possibilities. For each of those extracts, you also need to extract a clean portion of the swab in the same way. So here we've got layers of matrices. We want a clean swab that hasn't been exposed to anything. We want a swab that's been exposed to whatever the original matrix where you found these particles or stains were. For example, if you have a manufacturing vat that had sodium chloride solution, and then you found a residue on the edges that you wanted analyzed, well, we'd need a clean swab. We'd need a swab that had been dipped in sodium chloride solution, and then we would need the swab that had picked up the residue. And all of those are going to be treated in multiple ways. And each of those extracts is going to be submitted for analysis so that we can properly account for all of that information, if it's genuinely from just the residue or if it came from either of those matrices. Another very popular sampling device is tape. Regular old adhesive tape is a great device for picking up small particles. And when you've got something small on the surface of, say, a plastic tube in a manufacturing facility that you want to sample, grab a piece of tape, stick it on there, and peel it off. That's a pretty quick way to grab it, and you know it's not going to go anyplace. Well, unfortunately, this also picks up some things that you're not expecting. If you're looking at the image on the right, I'm assuming the particle of interest here is probably that nice long brown fiber, looks maybe like a hair, but we've found a whole bunch of other things too, this little red fiber and a whitish one and a black particle and little white pieces. I mean, these are probably all common environmental debris, just dust, but we don't know that when we look at the tape, and we have to determine which particle of interest is really there and where the others may have come from or not. Additionally, that tape then has to be brought or submitted to the laboratory for analysis. So you're either going to fold it over on itself and create a little tab, which is probably introducing some debris from the other side of the tape as well, or you're going to stick it on a substrate, like a piece of paper, and you have any of the debris that might have been on the surface of the paper, and those paper fibers are all becoming part of the matrix now and need to be considered when you're isolating your particle. It's not impossible or necessarily all that difficult to isolate a particle from tape, but it adds some challenges. You have to know exactly which particle you're interested in, and you have to be aware of the amount of adhesive. When we cut that out, you can see how much adhesive actually stays stuck to even a small piece of that particle. And this can be removed manually or with some solvents. Most adhesives are acrylate-based, and it's very possible that in subsequent analysis we're going to see evidence of an acrylate. And this would be reported as, we found this material and some acrylate, possibly from the adhesive, because we know it came from adhesive. But that might be erroneously excluding information if there is in fact an acrylate in your sample. Acrylic fibers, of course, are going to have acrylates in there, and certain other polymers as well. So you need to be aware of when the matrix is contributing information and when perhaps assuming it's from the matrix is not helping you. And the final challenge that I will discuss are some of those satellite containers. We often look at something, it looks really clean, and that's fantastic, and that's how we're going to package our samples to be examined later. When you take a really close microscopic look at these, they're not as clean as you think they are. Plastic Petri dishes are great to store things in, and if you look at those under a microscope, it kind of looks like it's snowed inside. Every surface where the plastic bottom meets the top, every time it touches against each other, it's shaving off these microscopic particles. And these run the risk of getting on any sample that is contained in the Petri dish. But likewise, any sample put in the Petri dish could be shedding particulate into the dish itself. So at the end, you don't know. You don't know if the particle stayed where you put it, if the particles from the Petri dish wound up on your sample, or vice versa. This can create a perfect storm of confusion for what you're looking for. The other satellite container that creates a lot of issues are sterile containers. Sterile means that it has been treated to prevent microbial growth. Sterile does not mean particle-free. And just as an example, this image that you can see here, I took one of our sterile 50 milliliter centrifuge tubes from the laboratory, filled it about half full with filtered deionized water, gave it a shake, and then filtered it in our clean room. And I found little particles and fibers. It's because the sterile container wasn't treated to minimize, particulate. When we have an instance like this and we report, here's all the particulate we found, and someone says to us, well, I was expecting fibers, but not these little black things, the first thing I always tell them is it might have been from the container. And if you tell me it's sterile, I'm going to tell you that that's not particle-free. We'll be able to identify what's in there, and the container itself isn't necessarily contributing any chemical information, but we're not going to always be able to source the original location of those particles, if it came from the sample or if it came from the container. So in that instance, your analysis may be compromised by using sterile containers. So now that I've explained all of these challenges and taken away all of your very favorite sampling devices, how are you supposed to approach this problem? Well, there are ways around everything, and sometimes, usually, knowing what has happened to a sample before we see it or before it gets to whichever laboratory you're using is the best defense. So call us. Call on our expertise for how best to sample or how best to package. Email us and let us know this is what we had to do. We didn't have any other options for how we were going to sample this. And we'll be more than happy to work with you to give you the best, most useful results possible, taking into account your matrix. This is also the time to talk to your chemist, talk to your project leader, and find out which samples they may need as a matrix comparison, because as we pointed out with the swabs, sometimes that might be something you're not automatically thinking of. I want to thank everybody for their time and attention. I know we have a questions box on the screen. Please feel free to use it, and I welcome any questions you have.
Thanks, Andrea. Yeah, that was interesting with the containers. As Andrea said, please go ahead and type in any questions that you might have, and we'll go through them here for a bit. I was going to ask Andrea, based on your last, getting towards your last slide there, do you recommend to your clients that they should rinse out some of these containers before they use them, or?
If it's possible, absolutely. We have the benefit here, the example I showed, we have clean room water that's been filtered in a particle-free environment. I mean, your water carried through the laboratory may pick some stuff up too, but by all means, it can be very useful to rinse your containers out a little bit before you start putting your liquid samples in there. It's better than nothing.
Yeah, at least it's a start. It helps.
And in that case, send us a sample of your rinse water as a matrix blank.
Oh, yeah, good idea. Okay, we have a question here from Anne. Are you usually rinsing the particulate coming from a liquid matrix?
Yes, we are. To the extent that we don't have to be concerned we're going to dissolve away our particulate. But when we filter, there's automatically a rinse with the clean room water that I've mentioned before. But frequently, if we know that those samples have come from, a protein-based drug, we'll go ahead and, as we're preparing them, do individual particle rinses with an appropriate solvent also.
Okay. Anybody else have any questions out there that you would like Andrea to answer? Here's another one from Anne. Are you performing the picking?
Sometimes, yes. The clean room is much better at it than I am, but we do see a variety. I am able to do some of my own preparation work, so I will spend about half my day with my face in a stereo microscope doing particle preparation, picking, including any of the rinsing necessary. Filtration has to be done in our clean room, though.
Okay. All right. Laura has a question. Can you recommend a particle-free sampling device such as loupes that are particle-free, or pipettes, perhaps?
I wish I could. Honestly, I have found pretty much nothing that's truly particle-free. The best thing to do, and actually the approach that our clean room takes when we're doing filtering and the like, is to rinse those items that you're going to use with cleaned particle-free water in a controlled environment rather thoroughly before you do anything. That's truly the only way you're going to get something really particle-free. Other than that, just out of packages, there's always going to be the risk of mild contamination.
Okay. Question from Mary Lee. Do you have microtome capability for foreign matter embedded in a solid matrix?
We do have microtome capability. We also, in some instances, have found that hand cross-sectioning is easier, and that is something with response to an earlier question on my particle picking. No, I'm not as skilled with that. But depending on the nature of the substrate, sometimes you can actually get a cleaner cut with a well-trained microscopist, but we do have a microtome as well.
Another one from Laura. Is there an advantage to using colored sampling devices to aid in differentiation between the sample and the sampling device?
Color can help. What we tend to do and use more to really make a particle stand out are different lighting conditions. Whenever we do our exams, we always look at our samples under oblique, transmitted, and coaxial illumination. And we'll find a lot of times that one of those may be better, or a combination of those may assist. Often we use colors to improve our photomicrographs, actually. We'll put a strongly contrasting color behind the particle to make it jump out. It helps. There are ways to work it if you don't have that option.
Okay. Mary Lee, do you use gold-coated filters for IR reflectance from the filter surface rather than transfer to sodium chloride window via a tungsten probe?
When we do our IR, we will use the mode of infrared that's appropriate for the sample. So if when we do our particle prep, it's going to be done in IR reflectance, we will put it on a reflective substrate. If it's something like a true fiber, which is 90% of some of the particles I always think of when I think of it, we'll do that by transmission. But that determination is what I would call more sample prep for analysis than necessarily isolating the particles.
Okay. Well, I think that might actually do it for the questions. And thanks again for attending today's Macron Group webinar.